Hello and welcome to today's webinar. My name is Mark Schrader and I'm an application scientist at Newomics. And joining me is Sophie Bianchi, a marketing specialist at Newomics. And we will be the moderators for this webinar. We're delighted to be hosting Simion Kreimer. Simon is trained under Barry Karger for his graduate education and did proteomics at Johns Hopkins University. And now Simon is the chair of innovation in the Van Eyck lab at Cedars Sinai. Simon, the floor is yours. Take it away.
Thank you very much, Mark, and thanks to everybody who tuned in. So as Mark mentioned, we're at Cedars Sinai, which is a hospital. It's a little bit of a unique place to do mass spectrometry research because in our sort of environment, I'm on the academic end of things where we do sort of fundamental research using high-resolution mass spectrometers like the Orbitrap and the TOF instruments. We do biomarker discovery. We have a special interest in doing deep proteomics, also high throughput. We do a little bit of assay development, but that sort of gets passed down to a whole other entity called Precision Biomarker Labs, with which our group is closely intertwined, where they really focus on developing targeted assays and validating them. And in turn, these will be passed down into our CLIA lab, which is still under development, in which we can take these assays from biomarkers we discover at the very academic end, and that have been kind of optimized in PBL, and then implement them in patient care. And of course, CLIA and clinical assays are very, they require kind of the highest quality of analytical rigor. So although we don't necessarily need this on the academic end, that's always something that's in the back of our minds. How can we do better reproducibility? Higher throughput is nice also. Robustness is very important, and most importantly, reproducibility and accuracy. So even in the academic end, that's something we think about. And that's part of the reason we started considering going to the microflow rate. So when I joined the lab, and all the graduate work I've done, as well as the work at Johns Hopkins, was mostly nanoflow, looking at 300 nanoliters or so per minute flow rates. It's sort of a standard in the mass spectrometry field. I think it's still the most widely used flow rate. And although sometimes you can take that for granted, if you start reconsidering it, you realize that it really impacts a whole lot in the LC-MS ecosystem. Everything from ionization efficiency, which impacts sensitivity, translates to depth of analysis, also relates to resolution of the liquid chromatography separation. Also relates to the column options. You can have different column bores, different media may not be available in every size you want. That, of course, bigger columns are more expensive, so that impacts cost. You think about the optimal amount of sample you can load, which then translates also into cost, because you have to consider the reagents you're using to prepare a lot or a little bit of samples. For us, the most important attributes are the robustness of the setup, which directly relates to reproducibility, and also the throughput. We like to be able to analyze a lot of samples quickly. So those are the most important attributes for us when we're considering flow rates. So for biological fluids, this makes a lot of sense. First of all, like plasma, especially a urine, whole blood, those things are not really limited in protein. And we also have to consider there's a high dynamic range, meaning that very abundant proteins sort of mask the lower abundant stuff. And so typically we expect limited depth in terms of how many proteins we can quantify. There's also a lot of different contaminants in biological fluids, things like salts, metabolites, lipids, protein fibrils that are not completely digested. And we have to also recognize that there's very high variability in things like plasma, even from hour to hour in the exact same patient. So we really need to do a lot of replicas to understand the true biological variability versus something that might be an artifact of sample collection or something like that. So in that sense, we want high throughput to analyze a lot of samples quickly. We want robustness, so our setup doesn't break down from all these contaminants. And we can kind of sacrifice sensitivity and depth because we don't necessarily want high numbers of proteins. We just want uh, proteins that we can quantify in every single sample to really understand it. Um, so in that sense, going to a higher flow rate from nano to micro makes a lot of sense. I'm kind of cover each one of these specific attributes in the next few slides. So when we think about throughput, a lot of people might say, well, I can run 60 samples per day or something like that. But in theory, you can run any setup with any gradient you want and achieve that result. What really kind of comes down to is the instrument utilization. So if we compare the two setups we frequently use in our group, we have our sort of nano-ish flow rate setup where we use a 2-meter micropack column and we run it at 1.2 microliters per minute with a trapping column. So with that setup, it takes about 15 minutes for the peptides to elute off the trap and get into and go through the 2-meter column and fly into the mass spectrometer. And then we also require an additional 20 minutes to equilibrate the setup. So out of the 140 minutes that we run the system, we get about 75% usable data. When we compare that to our faster flow rate system on the CapLC, it just takes 5 minutes for the sample to make it, for the peptides to make it through the trap and the column, and then 10 minutes of additional time to backflush and equilibrate the system. So we get about 79% utilization. At faster flow rates, these kind of sunken costs of dwell volume and reequilibration are lower, so it makes sense to run faster gradients. If we try this with the 2-meter setup, we will be using, if we try to run a 1-hour gradient on the 2-meter setup, we're already sacrificing 35 minutes just for the samples to get through and to fix the column, so the utilization is much lower. So it's not as good of an idea. So for throughput, faster flow rate is better. When we consider robustness, we can just do a pretty simple experiment. Consider what would happen if we add 1 microliter gap into our retention time, to our setup. So this can happen if fitting slips out or something like that happens. It's pretty detrimental at low flow rates. If you're looking at the very lowest, like 20 nanoliters per minute flow rate, that causes a delay of 50 minutes. At 300, it's still several minutes delay. But at 10 microliters per minute, it's negligible. So if such a gap is not really important, then we can do stuff like add trapping or guard columns to keep our system running longer and with higher robustness. We can also use wider bore columns, which are less likely to clog because there's a higher surface area. And we can add a lot of samples, a high amount of sample to these bigger columns, which translates to higher reproducibility. So again, the benefit is pretty clear that the higher flow rates work better in terms of robustness. The kind of downside to the higher flow rate is electrospray ionization efficiency. There's a great deal of data out there showing that the lower the flow rate, the higher ion utilization. So the sensitivity increases. This comes from the way Electrospray works. The theory is pretty basic. You have a potential between your emitter and the mass spectrometer. And when the peptides spray from the emitter, they come off as droplets that hold a charge. As the solvent evaporates from the droplets, the coulombic repulsion becomes too strong. The droplets burst into smaller ones. And those are as the solvent evaporates further, those burst again, making smaller and smaller droplets until the analytes, the charged analytes, are ejected and are able to be detected by the mass spectrometer. So if we start at a lower flow rate, the droplets start out smaller, thus more of them make it to the stage where single individual ions are released and are then detectable. So this theory works pretty well. We can get really high ionization efficiency at low flow rates, but for biological fluids, it's not necessarily important because we just have so much material we can inject a lot and still be able to detect it, even if our ion utilization isn't super great. The other aspect is we can kind of cheat the system with a multi-nozzle emitter. So if we have this 8 nozzle emitter that Newomics has, we can deliver flow at 10 microliters per minute, getting all the benefits of robustness and throughput. And then at each nozzle, the flow rate is essentially 1.25 microliters per minute. So in a sense, we should see an increase in sensitivity. Now, we didn't do too many head-to-head comparisons between like single nozzle, multi-nozzle setups. For us, kind of the adaptation of Microflow came at the same time as Newomics emitters. So we put them on, and we noticed immediately that the ionization was robust at the flow rate we wanted to work at. The spray was very stable, and the nice thing about having a lot of nozzles is even if one of them gets clogged or two of them get clogged, you still get pretty good signal, and you can continue running your experiment if it's inconvenient to swap it out or clean it. We did have one piece of data where we just directly compared the Newomics emitter to house DSI source or heated ASI source. And we just took a NIST 440 peptide standard, ran a 10-minute gradient of data-dependent acquisition, and we get more peptide IDs with the Newomics. And my colleague Annie did this work. But for the academic side, it was just really nice to have a solution that worked well at microflow rates immediately out of the box. There is a lot of data out there comparing these emitters head to head, so I encourage you to look into that if you're interested, to look into the sensitivity aspect of this technology. This is our basic setup for our microflow separations. So we use the Ultimate 3000 nano HPLC equipped with the capillary flow selector, or we can also use the microflow selector for this purpose. We use both valves. One valve is connected to the loading pump going through the autosampler so that we can inject the sample in one direction and then backflush the trap in a different direction. The second valve controls the trapping column. It either connects it to the autosampler or to the analytical pump, so we can then use the analytical pump to deliver a gradient to elute the peptides off of the trap and onto our analytical column. We use either the 300 micron ID by 15 centimeter Phenomenex Luna Polar C18 column with the 3 micron particles. And we found that in a lot of ways, it's equivalent to the Micropack CapLC column, which we also use. The thing we love about the Phenomenex is that they're really cheap. We can run them for a couple hundred samples, even if there's some contaminants in there. They just get used up, and it's not a big expense. The pharmafluidic column is quite the opposite. They're really expensive, but we get better separation with those, so we use those for special projects. And of course, we use the Newomics emitter, whether it's in the adapter kit or their special source. We typically run data independent acquisition either on the Orbitrap instrument like the Explorers 480 or the Lumos. And we now also have a timsTOF, which I will talk about in the last few slides. So when we developed this platform, we qualified it using whole blood samples. Actually, we qualified it on a lot of different biological fluids, but I'll just talk about whole blood for now. So the way we do this is we do a cursory measurement of reproducibility by taking five different aliquots of the same sample, and doing 5 different digestions, and then in duplicate we inject it over each sample over 3 different days. It's shown here. We get 4,700 to 5,000 to over 5,000 peptides. We also do just technical replicates when we inject the same sample over and over again, and that's also in that range in terms of peptide identifications. And then we also do the protein quantitation, looking at reproducibility. So for us, the kind of key thing is to have a CV under 20. We get over 300 proteins at that below 20 CV. Below 30, we get just under 400 for all 3 days, and then close to 400 for the interday reproducibility, meaning between all 3 days. So quite nice results. It's reproducible enough for us to work with, and so we're quite happy there. But we don't really get a good grasp of the robustness of the system unless we run hundreds of samples. And I'm going to present a study where we work with plasma and injected over 200 samples on the same setup and kind of cover how the data looked and the different attributes of the data quality. But before I get into that, I kind of want to discuss how we process data independent acquisition data. We have kind of a unique pipeline, actually with 3 different options, and that comes into play with the project we did there. So we can do this with either FAME separated DIA data, or in this case, I'm just going to present kind of conventional DIA. We have this data independent acquired data, which means we didn't just select individual precursors. We went through the whole mass range from 400 to 1,000 m/z, fragmented every single peptide because we systematically used big windows. So to interpret that, there's 3 pipelines we can use. So one is the AUTA library, which means we take the data files. We use an algorithm called DIA umpire, which essentially converts the data to data-dependent acquisition spectra, data-dependent acquisition-like spectra, and then we can use any kind of algorithm to search it, sequence, mascot. We really like MS Fragger. It's a free software. It's very fast. And then we basically do our false discovery rate, and then assemble a library from that. We also have the library-free approach, which is available with the software called DIA-NN. That's sort of our preferred platform at the moment for DIA data. It still requires a library, but when we do the library-free, it creates one out of peptide sequences using machine learning algorithms. So you just provide it with a FASTA file full of the proteins in your organism, and then it does an in silico computation of what the fragments, what the spectra should look like for each peptide, and then uses that as the library. Another approach is that we can do separate experiments to look at our data more closely, to assemble a library for our project. This was previously done with using fractionated samples, but we use what's called gas phase fractionation, where we focus on the small. Well, I'll go into that on the next slide. But basically, the benefits and downsides of these are kind of drawn out. But out of library, we tend not to get a lot of IDs, but we don't have to do any extra work, and we can add post-translational modifications into our search to be able to discover those. With Diane, with library free, it gives us the most identifications. We don't need any extra data. It's a little bit slow to create this library, and they have implemented some features recently for post-translational modifications, but we haven't really evaluated them yet. And for our own homemade gas-phase fractionated library, we get a little bit less IDs than the library-free approach, but we can be pretty confident about them because we've kind of analyzed these spectra before, and with our own algorithms, and we can put as many post-translational modifications as we like. So the library generation with gas phase fractionation works like this. So instead of doing any fractionation, physical fractionation of the sample, we just tell the mass spectrometer to look very closely at small segments of our mass range of interest at a time. So, for example, we could do 400 to 500 in one run, 500 to 600 in the following run, and so on. There are some really distinct advantages to this strategy. One is that retention time isn't influenced by changes in the matrix. So when you fractionate the sample, every peptide is now in a different matrix than it would be when you're doing the analysis. And the philosophy here is that you want your library to most closely resemble the samples you're going to be analyzing, how you're going to analyze them. So in that vein, the fragmentation energy is similar. We're sampling every single precursor. We don't get extremely higher sensitivity than our analysis, but that might be a good thing because we're not putting any peptides in that we can only see if we fractionate the sample. And that kind of keeps our library just the correct size for our experiment. So that's the strategy we selected to generate a library for this cohort. We also implemented a strategy called real-time quality control. So there is the idea that you could put QC samples in and kind of monitor those individually. But we can also use this tool from Biognosys called Quick. And as long as you have their peptide standard in the sample, the software will basically analyze any run you give it and spit out some very critical quality information about your runs. So some of the really important attributes, and here each graph is showing all 200 samples. I didn't include the axes, but basically each point is a sample in our whole 212 sample cohort. And we look at stuff like MS1 intensity at the very top left box. That's pretty stable. MS2 intensity is stable. Peak capacity is very important, so we can check to make sure our column is still performing well. And yeah, the range there is 185 to 210, so it's stable enough for us. The retention time of these peptides that we spike in, we have to make sure they're stable to catch any issues with the LC early on. The peak width, also very important to make sure that the quality is still good. And then it also does kind of a search and it tells you how many IDs you have. So it uses its own library, so it's not necessarily the same thing we would have if we're doing the analysis ourselves. But we might notice there's some dips in the identification. So those are samples we might want to re-inject. And this, in a way, it obviates the need to add quality control samples, especially for samples where every sample is really simple, like plasma, very similar, like plasma. I mean, we did 200 and they're very stable. So do we really need to add extra ones to check? The other thing is you will know if there's an issue pretty much immediately. You don't have to wait the 12 samples to run a QC sample. So it's kind of an interesting strategy we used here. It saves some time, some instrument time, and that's always nice because we're using the samples themselves as the QC. So we can then kind of combine our gas phase fractionated libraries, which we generated from pooled samples, and the library-free approach from Diane. We did the gas phase fractionated libraries two ways. One is with DIA, where we kind of do really small windows for a limited segment of the mass to charge range. And then the data dependent acquisition, where we still use the gas phase fractionation. We just look at pretty small ranges of the mass to charge range and then just do data dependent acquisition on that. Both of those approaches are quite complementary. We get 1,300 peptides that we don't see with the DIA approach, but the library-free strategy still gives us the most identifications. So these are basically how the peptides were identified, the distribution between the different search strategies. We can then combine them, and this is how the identifications look over the whole sample set. So we have different groups and different cohorts in this group. They all have to do with heart injury and heart attacks. I'm not really going to go too much into that detail, but the identifications are fairly stable. They're all around, between 5,500 and 7,000. And then the vast majority of peptides are in, well, over 3,000 peptides are in all the samples. And we also included phosphorylation as kind of a proof of concept that we can do these PTMs. And so quite a few phosphorylation sites are also detectable in almost all the samples. So this is an indication that we can do PTM analysis with this approach. And we could always add other modifications to our gas phase fractionated library searches if we wanted to. The protein level is where we're really interested in, and essentially almost 300 proteins are seen in every single sample, which is excellent. And then if we want to look at the different CVs, we have to keep in mind that this includes biological variability, which is much higher than instrument variability. But it's interesting to find that there's still a handful of proteins that are quite stable between the different, within each group. So even at CV40, which is high, but we have to consider that like the measurement, the fold changes we look at are twofold or more. So the CV40 is not really a big deal. There's like hundreds of proteins that are still very stable within each group. So we'd be able to pick up quite a few significant differences in this experiment. And that's what we're looking at here. So we have basically I've plotted all the proteins that are seen in every single sample. That's 274 proteins on the left, and it's basically just the average in each group, and then plotted by the combined average. And basically, there are quite a few outliers, even in this very stable set of proteins, which is encouraging because now we're looking at some real differences. If we start looking at the sort of more variable proteins, which show up in fewer samples, there's even more differences, but we kind of have to take those with a grain of salt because we weren't able to measure them in every single one. But of course, maybe they're just showing up in one group versus the other. I didn't want to put too many volcano plots up, so this is kind of a simpler way to look at it. So in that study, I hope I convinced you that the setup is robust enough to look at hundreds of samples without really any major issues, and so we're quite happy with that Microflow setup. Next, I'm going to talk about how we can really get into the throughput. So our goal right now is to be able to analyze a full 96 well plate in a day, which means we have to do 15 minutes per sample. And it can't be like 15 minutes and then plus the injection time, then plus the equilibration time. It has to be really 15 minutes. And the goal there is, well, we have two real applications there. One is to do high throughput analysis of plasma. And if we can get just 200 or 300 proteins that are very reproducible, and get PRM-like quality data for those, we'd be quite happy. We don't necessarily need to go super far in depth there, because we're kind of hoping that the changes we're looking for are in those higher abundance proteins or can be represented in those. The other application is doing perturbation analysis on cell cultures. So for example, if we have a cancer cell line and we try 96 different drugs, we want to be able to measure the effects, and 1,000 proteins or so is plenty, just to kind of understand where the mechanism is coming from. To do this, we still use our Ultimate 3000. We are currently using the capital C column. still using the Newomics emitter, but we decided to do this on the timsTOF instrument because the time of flight mass analyzer is very fast. So it should work very well with these fast gradients. The first thing we did was to kind of look at what we can do, what we can detect in plasma at different loads for the setup. I should say we're also operating this at 10 microliters per minute. So the gradient, so we tried different gradients, we tried different loading amounts. At 30 minutes, we get about 2,100 peptides in plasma. This is also with the data-dependent acquisition. So it's not necessarily the most efficient approach we can do, but it will give us a baseline to what we can do. But if we move down to 15 minutes gradients, we don't get that much less in terms of IDs at the 200 nanogram load. We can load a little less. The peaks are sharper, so that kind of helps us out. And we get a little bit less peptide IDs. The protein IDs are pretty stable. So we're kind of meeting our goal of running these systems at the microflow rate and at fast gradients. If we look at our heat map, this is a plasma. We still have a minute and a half where we're loading the sample, and then it takes about a minute for the peptides to get through the column and the trap. And so we get about 80% instrument utilization here. One thing we're going to do moving forward is we're going to be able to tuck this the sample loading step into the gradient. So we will maybe reduce that time and increase the utilization a little bit. So far with our plasma, we're now doing this with DIA at 15 minutes. We pretty stably identify 238 proteins. Well, 235 to 238 proteins, about 2,000 peptides. And in HeLa, of course, we get a lot more identifications, 2,500 to 2,700 proteins, tens of thousands of peptides. So for the cell, for the application of looking at cell perturbations, this is actually excellent. Because we'll meet our goal of running a whole plate, and being able to observe 2,000 proteins would give us a very clear picture of what's going on in the cells. For plasma, I think we can keep improving. We still want to get to about 300 proteins. The main kind of obstacle right now is figuring out how to handle the dynamic range with the time of flight. With the trap ion mobility setup. They don't like you to put too many charges on there, but with plasma you have these these highly abundant peptides that kind of throw a monkey wrench into the works. So we're actively working with Bruker and Newomics to sort of optimize this whole setup. But this is kind of, the future is bright with the setup, and we're hoping to be routinely implementing this 15-minute platform in the future. I would like to thank my colleagues at the Van Eyck Lab, especially Annie, for helping with the evaluation of the Newomics source versus the heated ESI source. Alexandra helped a lot with the initial qualifications of the Phenomics platform, that whole blood data I was showing, she helped put together. Chin and Ali helped with the Tim's time of flight instrument. And getting all that data, and of course Jenny, big thanks. The folks at Newomics, especially Pan, who helped us get going, Daojing, and of course Sophie and Mark for putting this webinar together. And at Brooker, we have Michael routinely helping us with the instrument. So big thanks to everybody there, and I will now take questions.
All right, thanks, Simon. That was great.